and PMW, participatory monitoring wheel, techniques, advocacy for the policy reforms at the government and intermediate levels in rural and urban developmental programs. Professor Sivappa sir has been effectively communicator with a strong relationship management skills and hands-on experience in networking with consultants, non-government organizations, government agencies, safeguarding the interest of the beneficiaries and stakeholders. Overall assessment and evaluation of the same reforms at government and intermediate levels in rural and urban developmental and project-based feasibility, financial viability, resource and partners delivery capacities, and ensure the quality delivery of funded project involving in-depth impact analysis of tasks, developmental program, proposals, documents, and training plans and materials, policy analysis and advocacy with media, government and non-government, international developments and subject experts. Professor Sivappa sir is an area of expertise. As I said, project management, research initiatives, team management, personal management, relationship management, monitoring and evaluation. And he has published the research publication more than 51 articles, book chapters of about 7, presentation of research papers and participation in seminar, conferences, short-term training courses, workshops, events, talks and lectures, altogether 50, and successfully guided 6 PhD students under the guidance of Professor Sivappa in the social work. And right now, presently, 5 research scholars are working under this able supervision, and major projects, programs are implemented. Total fund between the year 2004 to 2007 to 2014 is tuned up about 25 lakhs. Regarding the project of residual literacy, copper projects, collaborative programs with the University of Mysore and the Kingston University London, and successfully completed the major research projects as a part of the curriculum for the PG students. And Professor Sivappa sir is authorship of 2 books, Streams in River, A Journey into Inclusive Concerns, and translated a book, a Telugu play written Dr. Vijay Bhaskar, Hyderabad, into Kannada. And Professor Sivappa sir visited Denmark, Sweden, Norway, UAE, England and Nepal for the research collaboration and research and exchange program in collaborations with Dr. Wilson Mulya, Kingston University London, Dr. Venkatapula and Charles Stott University Australia, Dr. Frank Wittmann from the School of Social Work, Zurich University of Applied Sciences, Switzerland. Professor Sivappa is has a membership in the academic bodies of Board of Examination in various universities in Karnataka and other universities in India. And Professor Sivappa sir is an adjudicator of the PhD degree for various universities in India in his specialization, and a member of the Board of Studies in various universities in Karnataka and other states, and successfully organized about 12 international and national seminars for the benefit of students, researchers, organizers, stakeholders, and teachers under this able guidance. With this introduction, I welcome Professor Sivappa sir, and he accepted our invitation to addressing the gathering over here in view of the international conference, second international conference, organized, second international webinar organized by the Department of Sericulture Science under the chairmanship of Professor H.P. Manjunatha and all our associated teaching staff. I welcome you, sir, for this nice international webinar session Thank you so much.
Thank you.
So now let us move on to introducing our speaker. And I'm happy to introduce, okay, with a small request, I think there's a small change since our beloved sir is having another schedule at 11:30. So I'll introduce our speaker after our registrar is address the gathering. Sir, I request you to please address the gathering.
Thank you. First of all, I would like to congratulate the Department of Studies in Sericulture Science and Research in Sericulture Science. The staff members, we have taken the pleasure of organizing this timely webinars. This COVID has taught us many lessons, and the world has become, very narrow, and we are very close now because of this technology is put in place, and we are reaching through technology. See, Professor Dr. Hiroshi Kamamoto is from Japan, and he is with us, and many experts are located in different parts of the world. They are with us now at this moment. Only the technology has brought us together. I think maybe from that point of view, this COVID has taught us some lessons, and also this very good platform. And our university has geared up with a lot of changes, new changes for last 1.5 years. And all the department faculties, all the across the university, 74 departments are doing a wonderful job organizing this kind of seminars and webinars, and offline, online, both the dual mode, blended mode, and hybrid modes. So these kind of things are going on for last 1.5 years. So as a part of this initiative, university is trying to catch up with the needs of the students, academicians, practitioners, and all. And silkworm as a model for drug development, and this is one of the very important area of present times. They have come up with very good concept on this one. I think Dr. Hiroshi Kamamoto, Tokyo University Institute of Medical Mycology would enlighten all the participants in this regard. And Dr. H.P. Manjunatha, Professor and Chairman of the department, has taken a lot of pleasure in organizing this seminar. And moderator, Dr. Professor Jagdish Kumar, and also Professor Anil, Professor Sannappa, and also Professor Umakant have joined hands with Dr. H.P. Manjunatha. And always we are happy to assist, support in all respects to any department if they come up with any ideas. Our Honorable Vice Chancellor is extremely interested in reaching out to the people. Especially, we should encash this opportunity, these times, whatever the difficulties we have right now, and we should encash them from a positive point of view. That's what we are doing. And as I'm basically speaking on this particular sericulture and also the silkworm as such, coming from a rural background and also coming from a agricultural family, I would like to say a few things on this particular subject. I know when I was passing through childhood, we used to rear the silkworms in our place, and we used to put that food, mulberry thing for 3 times during the night time. 3 times we used to, we were waked up, and we used to feed this silkworm. I know that experience, and I come from a Polar district where it's famous for silk rearing, and silk, milk, and gold. That's a Polar district is famous for. So silk is one of the things, and so I know a little bit of this thing, but I may not be sounding technically very relevant to you. But I would like to use this opportunity to communicate with you. As Gandhiji quotes, to forget how to dig the earth and tend the soil is to forget ourselves. So this has got a lot of meaning, a lot of philosophy, and also farming as such. As we all know, about 70% of Indian rural households depend primarily on agriculture for their livelihood. Moreover, among different agricultural crops, sericulture has a long history as an agro-industrial activity for production silk, thread as a raw material for the textile industry. So, as most of us know, silkworms are growing for production of cocoons. During 2019-20, the raw silk produced in the country was about 35,820 metric tons and 11,142 metric tons from Karnataka alone. So you can see the contribution of Karnataka from this point of view. The sericulture and silk industry plays an extremely significant aspect of the economy and the social development in India. As I came to know recently, the usage of cocoons and silk does not merely confine to production of textile fibers and apparels, but there is much more. Since 211 AD, silk fiber has been used in the medical industry as surgical sutures. Whereas in the recent days, silk screws have been developed to fix bone fractures. Nonetheless, I came to know through while interacting with this learned faculty, Professor Manjunatha, Professor Jagdish, Sandappa, and Anil Kumar and Umakant, the silk is being extensively exploited for bioengineering and biomedical applications in the last few decades. Towards this, I am happy to mention that Professor Manjunatha has received a project from DBT Virac to develop a silk fibrian based bioprint to reconstruct maxillofacial defects, a noteworthy project in the University of Mysore. At this point of time, I am quite sure that, of course, most, as you all know, surprised that how could an insect silkworm is beneficial to mankind, which not only surviving the farmers, but serving the entire mankind in many ways. More so, as all of us know, in recent days, the whole world is facing a lot of health-related issues and a lot of research is taking place to develop novel drugs. And all these medical compounds have been tested using appropriate model organisms before subjecting to clinical trials. As I have come across, a stringent ethical issue acts as a limiting factor to test these drugs. So I came to know researchers are now looking for an alternative animal model, and interestingly, silkworm larvae are the one. In this context, I am so surprised and happy to state that silkworm larvae are in use not only to test the efficacy of drugs, but also to develop novel drugs, which is the need of the hour. You know, as a result of this type of investigations, novel therapeutically effective antibiotics have been successfully identified using the silkworm model. Towards this, Professor Manjunatha and his all colleagues in association with Japanese scientists, especially Amamoto, Dr. Hiroshi Kamamoto, they come up with a model system in India, which would be a good contribution to the biomedical field from both the institutions, especially highlighting our University of Mysore. So the present webinar is rightly organized and to create more awareness among the scientific community. Dr. Hiroshi Kamamoto from Japan is the right choice because he has got a rich experience in exploring the silkworm as a model animal in the field of drug discovery. So I hope all the students, faculty members, and academicians, researchers, everybody would get a benefit from this wonderful academic internationals across the world. So once again, I congratulate the Department of Sericultural Science in this regard. So let the departments take up this kind of things further with the different aspects. So we need to catch up. And also, I'm extremely happy that our Honorable Vice Chancellor used to congratulate this department. They have tied up with the farmers. That is a very important one. Reaching out to the fields. And it is our whatever we do, ultimately it is for the societal development and people's development, and people's living standards have to be increased from time to time. So in this context, the Department of Sericultural Science is closely associated, and they are having a collaboration with the farmer groups in and around Mysore. So I would, I'm extremely happy to share that one and let them continue this kind of new initiatives in near future. So I wish all the participants and also the learned academicians to interact one another and raise your questions and get the clarifications in this wonderful webinar. Thank you very much for inviting me. I wish all the best once again. Thank you.
So now I'm happy to welcome our Hiroshi Kamamoto for the presentation. Before that, I'll introduce him to all the speakers. In fact, Hiroshi Kamamoto is a faculty of, as you know that, he is in Kovid University. He has been working with Skimizu for a longer period. During that time, when he was working, I had a good opportunity to interact with him. And Dr. Hiroshi Kamamoto studied at Department of Pharmaceutical Sciences, Koishu University during 94 to 98 and graduated in 98-2000. He belonged to the lab of microbiology and studied on disinfectant resistant strains of Staphylococcus aureus and developed knockout mouse. And this is how he has been working hard to make the silkworm as a very good model system. Later, he moved to Graduate School of Pharmaceutical Sciences, the University of Tokyo, for his doctoral studies during 2000-2002. During this period, he established a system for evaluating the quantitative therapeutic effects of antibiotics using silkworm-based infection model. So this is the contribution which in fact led me to take up the research even in India, and one of my students, Dr. Kovid, is working with Staphylococcus mutans. After graduation, he worked as an assistant professor at the University of Tokyo, where he has been conducted research on the analysis of pharmacokinetics in silkworm and screened the therapeutically effective antibiotics which are derived from cyanobacteria culture supernatants using silkworm bacterial infection model. This is how the silkworm is not only considered as a bioreactor, but also he successfully developed the silkworm bacterial infection model. From 2005 to 2008, he successfully identified a novel antibiotics, that is lysocene E, through the discovery of antibiotics using a silkworm infection model at the Genome Pharmaceutical Institute. And this is how he navigated of using the silkworm to discover novel antibiotic, which has been attracted many of the scientists across the world, even at the industry's level itself. He then returned to the University of Tokyo as an assistant professor and analyzed the mechanism of lysine and found the lysine has unique mode of action which distinct from other known antibiotics, and therefore it is referred as novel antibiotics. Probably we have to encash this type of investigation to develop many more antibiotics. So now he is working as a professor in Tokyo University Institute of Medical Mycology, and he is engaged for the detailed studies on mechanistic analysis of therapeutic activity of lysocene E, and he is searching for new antibacterial and antiviral compounds. Whatever the experiments he has done, they have been published with a high impact factor referred journals, and he has got a good credit on it. And for more details, of course, he is available on his website, and you can also check it. So with this brief introduction, of course, I asked him more detailed information, but he has given me only the brief introduction. For more details, he is accessible on the website. With this brief introduction, of course, I welcome him for his talk by sharing his rich experience. Moreover, I am happy to record here. He is a good friend of mine, and he has been supporting our research by giving a suitable strategies to address the issues related to developing the silkworm model system in India. So I welcome you, Hiroshi, for this international webinar. Thank you. Now you can start with your PPT.
Thank you very much for introduction and giving me opportunity to present my recent work about the finding of the antimicrobial agents using silkworm infection model. May I start the presentation? Can you see my presentation?
Yes, we can.
Okay, I will start the presentation today. The title is Utilization of Silkworm as a Model for Drug Development. So I think that in the introduction, you also presented that the India also produce the many silks, and the silk industry is huge and large money. But in Japan now, the silk industry is going down. Very difficult to sustain the systems. But still, we have we can obtain the silkworm easily. So we can do the experiment using silkworm model. So today I will introduce the silkworm used as the animal drug model, and then the obtain. Sorry, this is not the slide shows. Okay. So we obtain these antimicrobial agents and then the antifungal agents and the lead compound from the antimicrobial agent. So using silkworm infection model, I introduced this model. I hope this model help the drug development for the Indians and the antimicrobial agents chemotherapy. At first, I will introduce what is the problem in the multi-drug resistant bacteria. So I focused on the multi-drug resistant Staphylococcus aureus. That this is still serious clinical problem. In the US, methicillin-resistant Staphylococcus MRSA causes the death more than 11,000 people per year. In Japan, also more than 4,000 people are dead, has been dead by the infection with the MRSA. But the newly developed antimicrobial agent for the Staphylococcus aureus is going decrease. Actually, until 2013, the new development of new antibiotics with new mechanism is decreased. So development of novel antibiotics is urgently required. So this is my newspaper report that in Japan also the share of the antimicrobial agent is decreased by the year by year actually. So the pharmaceutical company in Japan is not pay attention to develop the antimicrobial agent because of the low successful rate, and then it costs the huge developing cost. So the difficult to develop the antimicrobial agent in the mega pharma actually. Sorry, in case of the antifungal, the antifungal agent, the situation is the opposite. Antifungal agent is very small, so this variety are very, the compound is very small for the antifungal therapy. Only the 4 systems and then the 8 compounds are approved in Japan actually. So then this causes the less satisfaction and to treat the fungal disease. So we need to develop the antimicrobial agents and then the antifungal agent also. But the problem of the why difficult to develop the antimicrobial agent is only the few compounds show the therapeutically effectiveness. The most of the compound does not have any antimicrobial activity or antifungal activity, but even for the antimicrobial, the compound which show the antimicrobial activity or antifungal activity, or less than 1% show the therapeutic effectiveness. So we thought that if we can find that this therapeutically effective compound by the efficient way and rapidly. So it is easy. If there is a such system, so we can easily find the new mechanism, new antibiotics with new mechanisms. So we pay attention to the silkworm. So advantage of silkworm for the drug development, drug development is like written here. So the cost is very cheap. Compared to the mouse, the rearing cost is more than 10-fold lower. And then the silkworm is easy to rear. In Japan, we can purchase the silkworm as a egg. From this egg, we developed to the fifth instar. This cost is only less than 30 yen. So we can use the synthetic food like this. So silkworm is available all around the year in Japan. So this established system allows us to use silkworm as an earlier model. And the injection of sample also easy compared to the other alternative animal model like fruit fly or worms. They require the special equipment to inject the sample into the fruit fly actually. But at the silkworm, we can use the common disposable syringe to inject the substance into the silkworm. We can inject the lead silkworms, lead ink distribute into the whole body around the silkworm. So we can handle the silkworm because silkworm has large enough size to by the human hand. So the movement of silkworm also very slow compared to the other insect, because the silkworm is the highly highly adapted to the layering by the humans, so that's not necessary to walk around to obtain the to eat the food actually. That's the silkworm. The movement of silkworm is getting slower than the wild animals actually. So these two features allow us to inject accurate dose of the compound and then the high throughput screening. We can inject 300 larvae per hour by one person. In the drug discovery, the first kind of the law can be used for the silkworm. Usually, drugs screened by in vitro assay system, and then we confirm that their toxicity for the cell toxicity using the cell culture system, and then confirm therapeutic efficacy and toxicity in the rodent model, and then will perform a clinical study. Sorry. So here is taking a cost and then the time to find a good therapeutic compound and a low toxicity compound. So before testing the rodent model, we assume that the silkworm is a good model to find a better compound. So before rodent model, starting the compound that show the therapeutic effectiveness in silkworm model might be enhancing the drug development. Here, the summarize again the future of the silkworm as Asian model. The silkworm is the lower cost than mouse, then easy to obtain in Japan, also in I think India, and they have adequate size and low motility to handling the experiment, and then the less ethical issue. But these features allow us to use a lot of the individuals to screen the antimicrobial agents. So as sorry, we better to focus. I need to focus about animal welfare issues. So in Japan, there is the legal restrictions to use mouse. Actually, we need to apply the ethic improvement, and but recently very restricted, and we can use the more than 1,000 of the individual for the screening. But silicone is possible. Then previously mean the around the 1900 or something. So some of the antimicrobials are also antimicrobials are were screened by mouse. Its therapeutic activity, but at present, this system is prohibited. But the silicone is possible. So we need to care also. We need to also care about the animal welfare. So that in the one box, the mass individuals should be less than 5. But the silicone not necessary to care about this point. So we can handling a lot of the individuals at the same time. And the important point is infection experiments such as the negative cabinet is required for the mouse because it needed to prevent the infection from the mouse to human. But the silicone is not necessary. Not it is required, but not very cheap equipment is enough to prevent the infection. So these features silicone model can be used for the discovery by therapeutic peptides. In Japan, we have the National Biosource Center for silkworm. We can obtain the mouse, silkworm from here. This National Biosource project also distribute into the all around the world. So if you need some strain, please ask them to ask the center to obtain the silkworm. There's so many mutants exist in the so there's so many mutants exist in the this center. I'll discuss later about it. So silkworm drug discovery is addressing the cost and ethical issue. So we successfully identify the lysosine E. This is the actual lysosine E powder, very white and very light compound actually. So we established many disease model using silkworm. Today, I focus on the bacterial infection model and slightly a few I introduced the hyperglycemic model. And we established the other fungal infection model, so the innate immunity activation evaluation model using this silkworm, and it is applicable to identify the food such as lactic acid bacteria which induce innate immunity. And using a mutant strain is, sorry, it cannot move. Mutant strain registered in this biosource center. There is some similar. There is some disease model in this center, like the human disease. One or two is presented here. One is a sepialbutylin reductase deficient model. This is lemon lethal mutant in the silkworm. It can be applicable to use for the screening for the SPR deficiency or Parkinson disease. And second is the novel of the abnormal uric acid metabolism. It shows the phenotype of the oil silkworm. It can be applicable to screen the therapeutic compound for the goat remedies. So the Dr. Sezutsu group established the genetically modified the silkworms. So using this genetic modification, we established the hyperglycemic model and the evaluation, the model for the molecular targeted drug for the anti-cancer drug is established. This is the reported by the Dr. Matsumoto. Expressing the human type insulin receptor, it can be used for the anti-diabetic models. This not reported yet, but the expressing the cancer genes, it can be used for the screening of the anti-cancer drug. No response. Sorry, wait a moment. Sorry, the machine is slow. We established the bacterial killing model, bacterial model using silkworm. SRS, if when injected SRS into silkworm, silkworm killed by the injection with the SRS. So at the same time, if we injected the chloramphenicol antimicrobial drug, so the silkworm treated. Not only for the SRS, the variety of pathogens such as the enterococci and the pseudomonas aeruginosa, or E. coli, or Klebsiella pneumoniae, or the Candida albicans, or Cryptococcus neoformans, Aspergillus, and also fungi also kills the silkworm. And then we already established the therapeutic effective evaluation of therapeutic effectiveness of the antimicrobial agents. The silkworm model can be applicable to evaluate the virulence of the pathogenic bacteria. That will show that in case of the SRS, so we screened reduced virulence used in the transposon library mutant. We selected the 380 strains from this library, which are not assigned the function. Then the 27 strains are delayed killing of the against the silkworm. And the secondary, we found the 10 strains higher LD50 value, means low pathogenicity. We also confirm using the mouse model, so 8 strains reduced virulence. This is very high rate to find the actual virulence factors. Sorry, Dr. Manju, I think you will be present your infection model.
Oh, no, you carry on, please.
I think you will present.
No, you continue. You continue with your presentation. Yeah.
Okay, I continue the explanation. Sorry. Okay. Now it is better. So using this silkworm infection model, we established the quantitative evaluation of the antimicrobial agent. So I presented here that the antimicrobial agent used in the clinical. So this effective dose 50 therapeutic effectiveness per animal animal weight, and the dose is very close and similar value were observed. It means that the silkworm we can evaluate the antimicrobial agents using the silkworm infection model. Why we can evaluate the quantitatively therapeutic effectiveness of the antimicrobial agents. So silicone also has the pharmacokinetics like the humans. The pharmacokinetics of drug is consisted with 4 factors. One is absorption from the gut. The second is distribution into the organs. Third is metabolism, how they degrade the compound in the body. The fourth is the exclusion. Exclusion means how fast the compound eliminated from the body. So this pharmacokinetics in silicone is very similar to mammals. I show today for the example of metabolism of the drug in the silicone. This is the model compound 7-etoxycoumarine injected into the silicone. It was in case of the mouse, it was degraded by the P450, and then turned into the 7-hydroxycoumarine, and then the glucoconized, and then glucose is added in the compound, and then eliminated from the kidney in the mouse. Similarly, silicone is in the silicone, this compound is metabolized by P450, and then the decreased concentration by the time-dependent marmer, and then the conjugation reaction with the sugar is occurred in the silicone, and then eliminated into the pump in the silicone. So also, this result suggests that the silicone has the basic metabolic pathway, same as the mammals. I want to show the pharmacokinetics parameter of the antibiotics in the silicone. So clearance total is the elimination process, as reflected elimination process, and the VDSS means the distribution volume, suggesting that how the compound distribute in the silicone, in the animal body. These values are very similar between the silkworms and the mammals. This also suggesting that the silkworm has the proper pharmacokinetics like the mammals, so we can evaluate the therapeutic effectiveness of the compound, including the pharmacokinetics effect. We also evaluate acute toxicity of the compound using silicone. This result suggesting that the LD50 x-axis is the XLD50 in the silkworm of the compound. And the compound is listed in the OECD toxicity test compound. Then the LD50, the LD50 in silkworm also is well correlated with the LD50 in mammals. This mammals is a rat model. So the correlation efficiency is 0.72 is suggesting a good correlation where result. So the silkworm model can evaluate acute toxic effect of the compound. So I want to summarize the feature of the silkworm. The silkworm is suitable for evaluation of the therapeutic effect of the antibiotics, and the silkworm is low cost and less ethical issue, so we can use the thousands of labor for the drug screening per week. So pathogenic bacteria also kill the silkworm. Any human pathogenic bacteria kill the silkworm actually. The pharmacokinetics is highly conserved as mammals, and the toxicity of the compound also evaluated using silkworm. So these features allow us to screen therapeutically effective compounds using silkworm model. So we actually screen the therapeutic effect compound from the natural product. This is a result of the screening result from the natural product. I isolate the many soil bacteria around Japan. Actually, isolated the 15,000 bacteria as well, isolated from the soil and cultured, and the culture supernatant were tested their antimicrobial activity. Thus, 2,800 compounds showed antimicrobial activity against the SROs. So we injected all these supernatant to test the therapeutic effectiveness. So finally, we found 23 therapeutically effective supernatant. Then we purified the active compound from the supernatant. We finally found a new compound named lysosine E. Lysosine E consisted with the 12 amino acid and the short fatty acid chain. This structure is totally new, so we have the international patent and then the present into the 2015 as a paper. So antimicrobial spectrum of lysosine E is not, not so wide, but it can effective against the part of the gram-positive bacteria, including the MRSAs. Lysosine E effective against the SRS, the MRSA, and the Bacillus, and the Listeria monocytogenesis, and but not active against the Streptococcus species, but also effective against the Mycobacterium species. Mycobacterium bovis and Mycobacterium tuberculosis are also sensitive to the lysosine E. So it has potential to treat the tuberculosis also. Mistake. So lysosine E has the bactericidal activity. So this bactericidal activity is very strong compared to the other anti-MRSA drugs. So that please don't miss about this time is the minutes, not the hour. The less than 1 minute, lysosine E decreased the cell survival number less than the 0.01%. 99.99% of SRS are killed by the lysosine E within the 1 minute. The lysosine E has a very potent, highly potent bactericidal activity. I want to show this picture, but it looks not work. Sorry, this picture will not work, but I skip this one. So target, this feature is completely different from the known anti-MRSA drugs or other antimicrobials. Thus, we want to elucidate the target of the lysosine E. So we obtain the resistant mutant, then find we isolated the false resistant by using the mutagen. Then we found that the main A has mutated. So this mutant showed a slightly increase in the MIC and the decrease in the menaquinone content. Main A is involved in the menaquinone synthesis. Menaquinone is the cofactor of the electron transport chain. It is required to produce the energy production of the ATP in the bacterial electron transport chain systems. In case of the mammals, the mammalian mitochondria are using the ubiquinone, not the menaquinone, as a cofactor of the electron transport chain. So we assume that the lysosine E specifically interacted with the menaquinone, not for the ubiquinone. This result is suggesting that, and this result is the microcalorimetry analysis. If interacted with the substances, interaction with substances produce the energy, heat. So this small heat were measured using the microcalorimetry, but the dropped spike suggesting that the heat, suggesting that there is an interaction. In case of the lysosine mixing, in case of mixing with lysosine E and menaquinone, that heat was observed, suggesting that the lysosine E and menaquinone were interacted. But in case of menaquinone, there is no such heat. This is a dilution heat actually. So we confirm that the lysosine E interacted with menaquinone. So also we confirm, we want to confirm that how lysosine E kills the bacteria. We already know that the lysosine E impaired the membrane integrity of the SROS. So we use the liposome contain the menaquinone or not. In case of the liposome without the menaquinone, the membrane disruption activity is not so high. It's similar to the daptomycin actually, but in presence of the menaquinone in the liposome, the lysosine E efficiently disrupts the liposome. So we propose this mechanism that at first the lysosine E interacted with the menaquinone in the membrane, and maybe structure change or something that disrupt the membrane. Then bactericidal activity is observed appeared. Secondly, we try to show the therapeutic effectiveness in the mouse infection model. So very interesting phenomena were observed in this system. Therapeutic efficacy, antimicrobial activity of lysosine E is not so high compared to the other anti-MRSA drugs, such as the vancomycin, benzodiazepamycin is ranging the 0.5 to 2, but the lysosine E antimicrobial activity is 4. However, also, nevertheless, the lysosine E showed a good therapeutic effectiveness. Compared to the daptomycin, lysosine E showed a twofold lower ED50, suggesting that the better therapeutic effective activity. Why the lysosine E showed such good therapeutic effectiveness? So we found that the serum, addition of the serum, increasing the antimicrobial activity of lysosine E, which means decreasing the MIC of lysosine E against the SRS. Compared to the other antimicrovirals, the MIC is presence of the serum not so clearly different, but the lysosine E, what it is. Lysosine E clearly decreased the MIC. 60-fold higher antimicrobial activity was shown. But protein binding is also affect to the antimicrobial activity of the antimicrovirals. Higher protein binding usually decreasing the antimicrobial activity of antimicrobial agents. In case of daptomycin, it showed a highly protein binding, decreasing the MIC slightly. But in case of the lysosine E, protein binding is very high. More than 99% of the lysosine E bound to the serum protein. But however, the MIC is not affected by the invertly increasing the antimicrobial activity. So we want to identify the protein or compound in the serum, which increasing the antimicrobial activity of lysosine E. Using the at first we don't think that, this is a protein. So that's, I think we assume that this is a small compound. Thus we perform the ethanol extract and the ODS column chromatography. But actually this compound factor is a protein. The last purification step, the gel filtration, suggesting that apolipoprotein A1 and apolipoprotein A2 is correlated with the enhancing activity. We surprised that. So we also confirmed that using the recombinant protein, using human recombinant apolipoprotein were produced, and that we purchased also the human apolipoprotein too. Addition of these proteins also decreasing the MIC of lysosine E, suggesting that these proteins are enhancing the antimicrobial activity of lysosine E. So that's the high lysosine E showed a good therapeutic efficacy in the mouse systemic infection model, suggesting that maybe we assume that the apolipoprotein contribute to this good therapeutic effectiveness of lysosine E. We confirmed that this hypothesis using the mass deficient model of the apolipoprotein. When compared compared with the wild type, one is a vehicle, another is the lysosine E administered mass. The cell survival number in the kidney is decreased by administration of lysosine E. While in case of the apolipoprotein knockout mouse, this therapeutic efficacy is not observed. These results suggesting that the apolipoprotein A1 also contributes to the therapeutic effect of the lysosine E. This is the first report, the specific host factor enhancing the therapeutic effectiveness of the antibacterial agent. How to delete this one? Oh. We further elucidated the mechanism. Today, I not introduce the detailed order analysis, but we found that the lysosine E actually loaded by the menaquinone onto the cell membrane. Then disrupt the induce the small membrane damage at the sub-MIC concentrations, and then the lipid 2. It is required for the cell wall synthesis. This interaction is higher than the menaquinone. Thus, the complex is interacted with the apolipoprotein and enlarge the membrane damage and promote the bacterial cell activity of lysosine E. So we conclude that the lysosine E show the unique. Oh, sorry. It's okay. Lysosine E show the unique mode of action to therapeutic activity. This unique feature will be obtained by the host-based screening, like a silkworm. We also found that the silkworm hemolymph also enhance the antimicrobial activity of lysosine E. Thus, silkworm is a useful model to screen the host-adapted and the antimicrobial agents. Sorry, it's written in Japanese. I missed. We also found that the antifungal agents. This is the Astellas pharmaceutical group found that this compound collaborated with using our Aspergillus infection model. the name of the ASP2397, now Baikal, the BL2397. It also showed the good therapeutic effectiveness against mass systemic aspergillus infection model. This is when control, not administered this compound that kills aspergillus, kill the mass, but the administration of the ASP2397, they're completely survived, completely treated the aspergillus infection. So this is suggesting that the silkworm model is useful to screen the antifungal agent also. So far we identified these compounds using the silkworm model. Lysocene E is our identified compound. Nosocomycin is the Kitasato group. Dr. Tomoda identified this compound. It's also a new compound, again, effective against the MRSA. ASP2397 is an antifungal agent. So we also identify the antimicrobial lead compound from the synthetic compound, like the GPI0039 and the GPI0363. It's already published. The GPI3763 has a unique mode of action. It's targeting the SIG-A. SIG-A is the unpolymerase sigma factor consisting with the horo enzyme. And this targeted mode of action is different from the other unpolymerases targeting antimicrobial agents, such as the fidaxomycin or rifampicin. Also, we have the two unpublished compound. So we also established the other animal disease models, such as the hyperglycemic model. This model established by the Dr. Matsumoto, and now in the Meiji Pharmaceutical University. When feed the normal diet, the hemolymph of the concentration of the sugar in hemolymph is not so increased, but the highly concentrated, highly added the glucose diet, 20% glucose might be could be mixed with the food, so that in this the silkworm feed this diet, the hemolymph concentration of the sugar in hemolymph is increased. So we used this model as the hyperglycemic model. So in this model, the human insulin also effective. Administration of the insulin reducing the hemolymph sugar, suggesting that the silkworm also used for the screening for the hyperglycemic disease. So summary, we established the silkworm model can use for the screening of the therapeutically effective compound, such as the antimicrobial agent and the antifungal agent, or other disease treated compound. So in using this model, we identified the new compound, new antimicrobial agent, lysocene E. Lysocene E also has a new mechanism of action, and the target of menaquinone. Also, that this activity is enhanced by the host factor, suggesting that the silkworm is useful to identify the host adapted antimicrobial agents. Sorry, not the general guidance. So that this is acknowledgement. So we collaborated with the Tokyo University, the Department of Tokyo, University of Tokyo, Department of Pharmaceutical Sciences, the Inoue Laboratory, and then we collaborated with the Genome Pharmaceutical Institute. Actually, they screening the lysocene E, and lysocene E now perform the preclinical study collaborating with the AMEDO funding agency of the Japan Foundation for Medical Research Development. Thank you for the attention. I want to introduce, I am the associate editor of the antibiotics. The impact factor now increased this year, so please submit if you find novel antibiotics or related to the antimicrobial agent research. Thank you very much.
Highly informative and impressive presentation, wherein you not only revealed that Silicone is a good model, but you also shared your rich experience, how we can make use of the Silicone larvae, not only to go for screening of the drugs, but also you made it possible to develop antibiotics by using the Silicone larvae as a model. Of course, I'm very much happy to see that you have shown me the Tomica silk mill, where you can able to grow large number of Silicone larvae for biomedical screening and applications. So of course, you have highlighted the significance and better use of silkworm as a tool throughout your presentation, which is a valid and important model than any other organism. So you also showed how to use the silkworm larvae for screening of anti-cancer drugs. It is the one missing link as of now. Hopefully, India and abroad, we can able to go for using the silkworm larvae, not only to screen anti-cancer drugs, but also some of other drugs related to other human-related diseases. More importantly, whatever the drugs we are going to screen, the pharmacokinetics drugs is most important. How it is going to be work in the system of the silkworm is also important, which you have highlighted during your talk. Based on this, you made it the silkworm is a suitable tool to evaluate the therapeutic effects of antibiotics. Of course, we can use the silkworm to screen the antibiotics. Of course, we have done in our laboratory aspect to some of the experiments. But more importantly, as I understand and surprised, that you're using the soil bacteria for development of antibiotics, that is lysosine E, and how it is going to work with respect to other microbes. It is most important. So it is the field where we can able to look into the other resources where we can able to make use of the bacteria and make them express and develop the drugs suitable for the human-related diseases. So whatever we say, these antibiotic drugs has a very good actions, but we need to understand its mechanism of action. Wherein you proved it through your presentation that mechanism of action of lysosine is working very well in the silk. That's quite interesting, where we need to exploit this type of strategy even in India as well. So we know that the silkworm can also be used for a hyperglycemic model. So on and so forth, especially your talk is enlightened us with the recent developments in our laboratory as well as what we need to do further. So with these few points which I observed, I think now we open this your presentation to the audience for the interaction. I hope you can spare some time with us to interact with our audience, and I could ask, I could call upon the participants to raise your questions to Hiroshi Kamamoto to clarify your doubts. Please, sir. Please tell me, sir. Chugari, please sir. Unmute ಮಾಡಿ ಅವರು ರೈಸ್ please unmute.
Yes, sir. I am Mr. Chigri from Sericulture Department Extension Wing. Of course, I am a retired deputy director. So I am more interested in the extension aspect because whatever the results which are coming from your research stations, they are useful for the extension. Sir, before I ask the question, I am thankful to the University of Mysore and Sericulture Sciences for arranging such programs. Really, these are very useful. Since we are retired, we are getting ample of time to go through such sessions. First of all, I thank you very much for all of you for taking the trouble and also making it successful. And second thing, Dr. Hiroshi Kamamoto Sensei.
Yes sir.
Dr. Hiroshi Kamamoto Sensei.
Yeah, he is there. Please ask him.
So, are there any results? Of course, now we are using these microbes. Earlier, there was a book on sericulture that Silicone as a laboratory tool, but now the same tool is used for these microbes and other things. So, are there any results from the University of Matsumoto that fungal and these bacterial and virus resistant strains cannot be totally resistant? Any strains developed by using these microbes? This is my question, sir. Thank you very much.
Hiroshi, can you answer?
Sorry, I clearly not catch a point about this your protein. You ask question that you ask as a virus problem.
Yeah, virus and bacterial resistant variety. I mean races. Are they developed in Matsumoto Center?
Okay, for the bacteria, it is possible to evaluate the pathogenicity of the pathogenicity or drug therapeutic efficacy against the drug-resistant bacteria. But in case of virus, the virus is a little bit difficult because the virus highly selectively to the host. Actually, human virus cannot be infected with silkworm. So we need to use the silkworm virus. Actually, we established the method using the BMMPV. I forget the full name, but the silkworm virus. Using silkworm virus, and we evaluated the gancyclovir or aciclovir. It's useful for the herpes simplex virus treatment. With that, we can also evaluate the antiviral agents using the silkworm virus, but the other virus such as the RNA virus, we could not establish yet.
Thank you, Sensei. Do you have any action plan in future to develop any antibiotics for the silkworm diseases, Sensei?
So in future, I also developed the other. We found also other antimicrobial agents which has a new structure. So the other lysosine E also we focus on the further development. What are the other candidates also exist? It is okay.
Yeah, Sensei, in Matsumoto Station, other than silkworm breeding, are there any activities like your experiment right now? Because I visited in 2003, only they were concentrating on silkworm breeds. What are the activities now, Sensei? Matsumoto Center.
Matsumoto Sensei?
Yeah.
Matsumoto Sensei, you met the Matsumoto Sensei previously?
Yes, sir, 2003.
I understand. He is now the Meiji Pharmaceutical University. Like he is also continuously working on the disease model using silkworm.
Thank you very much. Thank you.
So, YSR ready, sir? Nirmal Kumar, sir, thank you.
Thank you very much.
Sir, ready, sir?
Sir, unmute ಮಾಡ್ಕೊಳ್ಳಿ sir. ಭಾರ್ಗವ ರೆಡ್ಡಿ ಸರ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ಸರ್. ಅಲ್ಲಿ ಅನ್ಮ್ಯೂಟ್ ಅಂತ ಬರುತ್ತೆ ನೋಡಿ ಸರ್ ನಿಮ್ಮ ಹಾ ಸರ್.
Can you hear me now?
Yes, sir.
As you know, the blood sugar in the case of silkworm is trehalose. I don't know how good is the testing of the drugs for hypoglycemic effect in silkworm, because major blood sugar is trehalose, not glucose. Glucose is produced only at the level of the tissues by the trehalase activity. The circulating sugar is trehalose.
Yeah, I understand that your question. The trehalose is the main sugar in the silicone hemorrhage. Actually, we know that, but this trehalose is converted from the glucose, consumed, absorbed glucose. So that's that this concentration also strictly regulated by the homeostasis, using the same system with the insulin-inducing signal pathway in silicone also. Thus, even if the trehalose, these differences exist, but we can evaluate the hyperglycemic, insulin-like drugs using the hyperglycemic model. Actually, we measure the trehalose concentration in the hemorrhage.
Yes, next question, please.
Doctor.
Yeah, please. Relocation, please, madam. Unmute, my colleague. Please unmute yourself.
Oh, yes, sir. Yes, sir. Sir, can you hear me now?
Yeah.
Yes, sir. Thank you, sir, for inviting me to attend this very interesting webinar. And I congratulate Professor Hiroshi for a very nice talk. My one very simple question to you, sir. When you test the effect of the antibiotics, what physiological age of the larvae you consider the most? Because to my opinion, the insect hormones, especially the steroid hormone, 20-hydroxysteroid, this might alter the efficacy of the antibiotic.
I understand. We don't know actually, but some steroid drugs looks effective against the pathogenicity of effect against the pathogenicity of the virulence of the reservoirs actually. And the administration of the steroid enhancing the pathogenicity of the microvirus. I don't know about the 20-hydroxysteroid, 20-hydroxysteroid, but the human applicable steroid is also work in the silkworm.
Okay, sir. Thank you, sir.
So,
and what physiological age you consider? Which larval age? Fifth instar, which larva?
Which larva?
Fifth instar larva, which days of the larvae?
Fifth instar larva. Yes.
Early instar or late instar?
Sorry?
Early fifth instar larva or late fifth instar larva?
Early fifth instar larva. Actually, we use the after immediately molted larva fed one day, then use next day.
Okay, sir. Thank you, sir.
Subramania, sir.
Good afternoon, Hiroshi Sanse, and also good afternoon to my colleagues, Professor Manjunath and Anil and then Sadapa. And actually, thank you very much for your detailed narration about how silkworm is useful in drug development. I remember the earlier days we were using silkworm as a model organism for genotoxicology, radiation biology, and many more. Now, you have introduced the concept that silkworm can be used in the development of antimicrobial, antifungal drugs, and etc. Very good information and very useful to the students community also. But now I have one question. Is there any possibility of using silkworm in COVID-19 vaccine production?
Vaccine production, I understand, yes. I think possible. The production of the protein in the silk gland is possible. The Dr. Sezutsu group can be modified, so genetic modified, they develop the genetic modified silkworm. If produce the spike protein in the silk gland, it can be easily to purify this spike protein.
Yeah.
Protein production would be easily to
Easy.
but the development of the silkworm individual might be have a take time. The
Yeah.
other way is using the insect cell lines. So the Japanese pharmaceutical company tried to product their spike protein, spike vaccines using the baculovirus infection cell line. Now.
Yeah. Good. Thank you.
Thank you very much.
Thank you.
Okay.
Any audience? Nirmal Kumar sir?
Very good afternoon. It was a very nice and lucid presentation which contained a lot of information. I compliment you for that. Any commercial application has been done because you have been working from 2015 onwards and you have published number of papers also. Any commercially people have taken this commercial application? Hello?
The lysosine about lysosine.
Yes.
We are trying to develop for the clinical application now. But now until the preclinical test was done. So next step, I think.
Oh, not next step. You are going to do that.
Yeah.
Okay, because this information generation is fine, but ultimately the logical, it should reach the logical end. That's what I personally feel, so that the humanity or the other, wherever it can be used, both antibacterial and antifungal drugs.
Yeah, actually, I think development is very important. We will try to develop.
Okay.
Clinical phase.
Okay, we look forward for that.
Thank you.
Thank you, Dr. Manjunath.
Thank you, sir.
So, Dr. Abid, you want to ask question, Abid?
Yeah. Good afternoon. Yeah, I am very indebted and thankful to the organizing committee. But actually, I could see some of my teachers in the webinar, Professor Vaishnava Reddy, Professor Subramaniam. Thank you so much. My first question to the speaker, Dr. Hamamoto, would be, as you discussed about antibiotics like losocomycin, lysosine, yeah, as per the therapeutic effects of this goes, have you tried them in humans in your lab? This is first question. One more question is, since with regard to the hyperglycemic effect, anti-hyperglycemic effect you were talking about, 1-deoxygeromycin is considered as potential alpha-glucosidase inhibitor. Have you tried that?
Okay, the first question.
Yeah.
Lysosine is still preclinical, not tried yet for the human. The second question that I think Dr. Matsumoto should be answer, but he's not here.
Okay.
The nojimycin also, I think, effective against the hyperglycemic model, but I don't know. Sorry.
Okay. What about the 1-DNJ? Has your lab worked on this aspect? As we can see an appreciable amount in silkworm excreta as well as in the silkworm larvae itself.
Sorry, I can catch up.
Okay, sir.
Sorry.
Next question, please. Professor DM. Sumne khud committee de rahe. Yeah, please come on screen.
Thank you, Dr. Hamamoto, for a very nice presentation on the development of drugs using silkworm as a model. And you actually used Staphylococcus aureus as the organism against which you have tested and how it has effectively contained the infection in silkworm. This is what, but by and large, what we see in silkworm, it is chiefly the mixed infection we see, not a particular infection alone. This because this you have done in a laboratory, infecting the silkworm with Staphylococcus aureus, and you have tested the efficacy of lycosine, and it has been found very, very effective. But when because the situation in silkworm is different, when we rear, it is predominantly mixed infection. Of course, when mixed infection is there, the expression of the symptoms of a particular pathogen only will be expressed, but other pathogens will be supporting the infection of other pathogens. So when such being the case, how do you anticipate or expect this particular drug what you are interested in testing would be effective? And as Dr. Nirmal Kumar just now pointed out, the commercial aspect of this particular drug. Because when you commercialize also, you have to look into the effectiveness when there is a mixed infection. Because you have Staphylococcus and Streptococcus type of this one bacillus group of pathogens, and sometime IFV, DNV, and NPV, various things are there. It is not exclusively a single infection. So will you please highlight on this particular thing with reference to the effectiveness of the drug, what you are, the drug in question?
Thank you. We don't assume that the mixed infection actually, but I think it will be show therapeutic effect against the mixed infection infectious situation if the lysosine E effective against these pathogens, I think, because the lysosine E is physiologically interacted with the menaquinone and then disrupt the membrane immediately physiologically. It's not require the pathogenic response. Just if the lysosine E effective against these microbe pathogens, it will be effective, I think. But we need to elucidate, we need to check, evaluate the effectiveness in the mixed infection status situation. I understand. Thank you very much.
No. Do you anticipate any elevation in the resistance of this particular silkworm against the viral infections? Because this is a drug chiefly working, exclusively working against bacterial pathogens. But does it provide any prophylactic effect against or action against the viral infections? Because you are containing the bacterial infection, you are keeping the bacterial infection at its lowest level, and thereby whether it also can confer some resistance against the viral infections.
Sorry, resistant viral infection. We don't know actually. Sorry, I don't have any aspect actually at this point.
Sir, Sarashodra sir, good morning. Nice to see you. Unmute not calling.
Thanks, sir.
Nice to see you, sir. After long time.
Nice to see all of you, sir. It's a very good opportunity. Today, our friends have kept this in our WhatsApp group. So I thought I will join because it's almost nearly 8 years since I left CSB and I moved out of Bangalore. So it's a nice opportunity. It's a nice opportunity to hear the lecture of Professor Hiroshi, given me this opportunity by the University of Mysore, and I think he has given very clear picture of what he's done. Maybe sometimes we are thinking no too much, because now he has done a laboratory work how silkworm is to be used, and there his duty ends. The next group maybe they have to see what if suppose some molecule is identified as a useful thing for controlling any disease or something. Maybe that has to be taken by another group using higher animals to test them, or ultimately to the human beings as a as a volunteers, no, as they did for the COVID test and other things. Maybe straight away taking this for COVID experiments, I don't think it will be so quick. But anyhow, it's a good thing that is useful at a laboratory level. Basically, we have so many problems in our this thing, in our what we call rules and regulations. We cannot use every animal as an experimental animal. So there, he has already explained in the very beginning that they can't use rats or other animals like as much as we can use the silkworm. So he has seen the convenience of silkworm as a laboratory tool for testing these medicines or chemicals, or what you call that, basically molecules. I hope that we'll get something better out of it for a period of time. It's very nice to see all my friends here. Thank you very much. Thank you for the opportunity.
Namaste, sir.
Namaste.
Sir.
Namaste, Randy.
How are you, sir?
Fine, fine. How are you?
Thank you, sir. Thank you.
Thank you. Thank you.
Any questions from the audience, please? So anybody is there? So yeah, somebody's coming up. Okay,
Hello.
Yeah. Please. Hello.
Sir, my voice audible, sir?
Yes.
Introduce yourself.
Sir, myself from Dinesh from Vijayawada, Andhra Pradesh, sir.
Please. Yeah.
I'm a student, sir.
Yeah.
Regarding fibrin market, silk proteins.
Okay.
Anybody?
You focus on the silkworm model system because the silk fiber and everything is out of our the theme of presentation today.
Okay, sir. Okay. Sir, thank you, sir. Thank you.
Okay. Next. So, any questions? Any more questions? Asha, are you there? Yeah. So, I think if there are no questions from the audience, I think I thank Hiroshi Kamamoto for your nice presentation as well as nice interaction with the participants of presentations. Those the senior professors are also raised there some questions and you clarified them very well. So I request my colleague Dr. Sanapa to say a word of thanks at the end of this webinar.
Sir, sorry for interrupting. Can we get the information, this PPT information, sir?
Yeah, please send an email. Probably I can forward this.
Yes. Thank you, sir. Very kind of you, sir. Very kind of you.
So thank you, Professor Kamamoto, sir, for giving an opportunity to propose.
I see that it is coming on YouTube also. Maybe
Yes,
it is recorded there.
Yes. In the University of Science, I request all the participants, the thing is available, especially the video is available in the university website. You just type it YouTube and the title of it, you will get it. And in today's data also, if you put it, you will get it. That is not a problem at all. But meanwhile, if you send your email, you will get the things from the Yamamoto and we'll forward it to you. That is not a problem at all. And if there are no questions, I thank Dr. Hiroshi Kamamoto, scientist and professor from Tokyo University Institute of Medical Mycology, and the professor has in detail explained about the silkworm as how exactly, as the people know that, including scientists and including layman says that only silkworm can be used as a cocoon production and also the meager byproducts of other things. But how exactly this can be used as a model organism for development of drugs? That is the need of the hour for everybody. Professor T. Subramanian also highlighted about whether silkworm can be used as a model organism or to test something for COVID-19. That is the future endeavor. So I thank our Honorable Vice Chancellor, Professor G. M. Kumar, for supporting all our academic endeavors in our department. I thank Professor R. Shivappa, just Sir has gave about detailed information about how exactly we are organizing webinars in the department or elsewhere in the university. I thank profusely Professor R. Shivappa for coming over here and having served as a chief guest for this particular international webinar on silkworm as a model for drug development. I thank Professor H.P. Manjunatha, organizing secretary or convener of this particular international webinar, for having conducted over this one. I thank Dr. T.S. Jagadish Kumar, professor. He served as a moderator for this particular international webinar. I thank Dr. Anil Kumar, Dr. Umakar, and all my research scholars, all the non-teaching staff of our department, and all the faculty of University of Mysore. Especially, I have to thank each one of you, Professor Y. Srinivas Setty sir, Veera sir, Dr. D. Manjunath, Dr. Subramania, and Nirmal Kumar sir, and also Chigri sir, and all others, and Vashini madam and Keshan, all Bela Keshavan, and all the scientists of across the globe, scientists, faculty, research scholars. Especially, this is very important for young budding scientists like research scholars, especially from all over the part of the world. I thank one and all for attending this particular international webinar. Keep attending the things we are going to send the information to you. And one more thing I want to request all of you, and we are in a position of giving the certificates, please, and we have already placed the information, that is this one, and just click onto it and you can submit your information to this chat box, so feedback, so that we can able to give a certificate for you, that is the certificate. I thank one and all. If I have missed your name, please excuse me. And Dr. M. Raju, from professor from Agriculture University, Bangalore, and Chandrasekhar, Professor S. He is professor in the Department of Agriculture, and also Chikkalingaya, and many other scientists and others. I thank one and all for participating in this international webinar. Thank you very much, and see you the next time. Thank you one and all.
Thank you, sir. Thank you.
Thank you. Thank you. Thank you very much.
Thank you, Hiroshi.
Thank you.
Thank you.
Thank you, sir.
Thank you all.
Thank you, sir. Thank you, sir.
Video.
ಬೇಗ ಸರ್ಟಿಫಿಕೇಟ್ ಕಳಿಸ್ಕೊಡಿ.
Thank you, Subramaniam sir. Thank you, sir.